Digestion of carbohydrates. Firstly, starting with the definition of digestion. Definition is nothing but breaking down or disintegration of these complex food particles into smaller food particles. The complex ones are broken down into smaller particles. How they are broken down? By the action of various digestive enzymes. Now, with the help of these digestive enzymes, the complex food particles are broken down into smaller. Now, why is because the smaller food particles are ultimately are maximum absorbed. They are absorbed properly, and also they are ultimately utilized for the energy in the body. So coming to the dietary carbohydrates, in the diet that we consume, we have polysaccharides, we have disaccharides, and we also have monosaccharides. Polysaccharides, example, starch, glycogen, cellulose. And then disaccharides, which is sucrose, which is a type of table sugar, and lactose, milk sugar, maltose, and monosaccharides, glucose, fructose, galactose, which are the different types of dietary carbohydrates. But only the monosaccharide form is absorbed by the intestine. So all these complex carbohydrates have to be broken down ultimately to monosaccharide so that they get properly absorbed. So digestion of carbohydrates mainly involves breaking down this big starch molecule into oligosaccharides, which is further broken down into disaccharide and further into monosaccharide, because monosaccharide is the only form that is absorbed by the intestine. Two different types of enzymes are required for carbohydrate digestion. Amylases, which mainly break polysaccharides into disaccharides, salivary amylase and pancreatic amylase, and then disaccharidases, otherwise intestinal enzymes which break disaccharides into monosaccharides. Example, maltase, sucrase, isomaltase, lactase, and trehalase. Firstly, digestion in the mouth. Digestion starts in the mouth with the help of mastication. So during mastication, the salivary glands secrete this alpha amylase. Now this alpha amylase, this is the starch molecule, which is a polysaccharide. So the structure of the polysaccharide can be linear or can also be branched. Now the linkages which are present in the linear are called as alpha 1,4 linkages, which are present in the branched, they are called as alpha 1,6 linkages. So salivary amylase mainly breaks this alpha 1,4 bonds which are present in the straight chain, and they hydrolyze the starch into a mixture of dextrins, maltotriose, maltose, and isomaltose. So this is the starch or glycogen molecule which has a large branched structure, and the bonds which are present in the linear structure are called as alpha 1,4, and the branch which is present here in the branch is called as alpha 1,6. So this salivary amylase mainly breaks the alpha 1,4 linkages to form a dextrin, maltotriose, maltose, and isomaltose. Now digestion in stomach. In the stomach, carbohydrate digestion temporarily stops. So the food in the form of bolus reaches from the mouth to the stomach. And in the stomach, because of the presence of the acidic gastric juice, this gastric juice has acidic pH, and that inactivates the salivary amylase. So digestion of carbohydrates temporarily stops in stomach. Next, the food reaches the small intestine. This partially digested food from the stomach will reach the small intestine. So while reaching the small intestine, the gallbladder bile secretes bile, and pancreas secrete the pancreatic juice. So there are two phases of intestinal digestion. First is digestion due to the pancreatic amylase, which is present in the pancreatic juice, and then digestion due to the intestinal enzymes, which are otherwise called as disaccharadases, like sucrase, maltase, lactase, and isomaltase. So this digestion due to pancreatic amylase, it hydrolyzes the alpha 1,4 glycosidic linkage, and further it breaks the dextrins into short oligosaccharides and disaccharides. So these alpha 1,4 linkages are again broken down by the pancreatic amylase to form short oligosaccharides and then disaccharides. So further digestion of these short oligosaccharides and disaccharides occurs at the jejunum mucosal lining with the help of the enzymes disaccharadases, which are otherwise called as intestinal enzymes. So in the jejunum, the brush border cells secrete these disaccharidases to give corresponding monosaccharides. For example, maltase acts on maltose to give glucose and 2 molecules of glucose. Isomaltase acts on isomaltose to give again 2 molecules of glucose. Sucrase acts on sucrose to give glucose and fructose. Lactase acts on lactose to give glucose and galactose, and dextrinase acts on alpha limit dextrin to give glucose and maltose, all of which are nothing but monosaccharides. So ultimately the large polysaccharide molecule is broken down into single monosaccharide units. So the end products of carbohydrate digestion are only monosaccharides. The end products of carbohydrate digestion are only monosaccharides like glucose, fructose and galactose. So to summarize the digestion of carbohydrates, the food that we are eating has starch or polysaccharides. So in the mouth, by the action of the salivary amylase along with the mastication, this amylase acts on the alpha 1,4 linkages of the starch molecule to form a mixture of dextrins, isomaltose and maltose. Now this long food bolus reaches the stomach where the digestion temporarily stops because this pH inactivates the amylase, salivary amylase. Now this food bolus again reaches the intestine. In the small intestine, the pancreas secrete the pancreatic amylase, which again dissolve the alpha 1,4 linkages to ultimately form the monosaccharide unit. And the disaccharides that are present in the intestine food are directly absorbed into the intestine, where they are acted upon by the disaccharadases or the intestinal enzymes to give corresponding monosaccharides. Cellulose, if it is present in the diet, it is not digested and it is excreted as such, which gives bulk to the stool or helps in digestion. Thank you.